You're Asher.
That's gonna get.
I'm James. He's Asher.
No,
He's Asher. No, I'm also James. He's obviously.
Yeah.
Wait, are you first?
Okay, you guys got about 3 more minutes. Let's see what you have. And then we're done.
Observation, Sherlock.
Don't take the mask off.
You know how to do it.
Yeah.
The mask off.
Yep. Yay.
Yeah, Miss
Very
Danny,
good, yes.
this isn't Dre.
Yeah, James doesn't change it. Take this back. That will help.
He's got his clothes hanging right down. If you value the dryness of the. Said it.
Oh.
Wait, there you go. I just said it now. Here you go. Here you go.
Annabelle, it's good. Hang on.
If you, okay, good.
Close.
Yeah.
Unbelievable.
What's missing?
By the way, did any of you lose sunglasses? There were sunglasses. Oh, you found them? You lost them? Yeah. They're in the middle of the front yard. It was like, I bet it belonged to somebody.
See that picture. I have no idea these.
How did you make it?
You know, it's just back with it. Whenever you're ready.
I go in there going to see the.
5 minutes.
Where some of that all down the side of it.
Stereotype.
Yeah, we got onion in there.
Let me see what happens then. Each.
Thank you. I need to see.
Where do I put my?
Oh, yeah.
That's.
Yeah.
Are you first head? Are you first head?
A little bit.
Are you nervous? Yes. You can bring them in. But it's mine. I got it. It's only you guys did.
Thank you.
Now 6 months, you can go past places.
Yeah. Yeah.
Actually,
Wait,
what?
One, and then you do some like else.
I really will. Don't sit cover for 5 minutes.
I really will.
It's already cold and shit. I'm.
Alright, wipe up that section.
5 minutes.
Why do none of them look? No, it's not that they put things on it.
Alright.
Just get ready.
So you my advice.
That makes sense. Neither have I.
I tried. You study. You turn it out.
You let that touch it there. Thank you.
After going out, we tried to make sure. Look at. It's not about like everyone's going with this small. Every person who's trying. Oh wait, that. We still from here.
Say it in your head, right?
Remember what you told me one day?
Here, and measure.
This is the one that. Oh.
All right, let's start. If you got stuff out on the counter that's trash, please get rid of it, or at least like collect it into one bin so that we can clean up space here. I don't think anybody wants to work on so let me clean as much space as I can clean. You don't understand your life, I don't think.
Why for 20 milliliters of ethanol? Oh, wait. Yeah. Oh, you could just use
Whenever you're done with all this, clean up this space here and leave all this stuff in and stick it over there, okay? So that it's out of the way. We gotta get going. Alright,
If you left it, you made it.
Next week is reviews.
Yeah, that's
what.
Oh. Are we done?
Yeah.
alright, there's a couple people still wrapping up, but they're gonna do that quietly, right? Right. We need to go ahead and finish our last lesson. Last lesson, next week is what? Review, and the week after that is? Finals, right, so you will, yes. Deoxyribonucleic, very good. So you're gonna need to, this week, you're gonna need to study for your Unit 9 test, because that will be due next week. And then you will roll in here and it's just exams and final projects, right? Right. You will have a special assignment about pizza,
Yeah.
right? Just like you did
That's
before.
what that was.
Yeah, be sure you go in and do that. It's easy peasy. All right. So I'm gonna go ahead and start. If you're still working at the counter, go right ahead, but it looks like the spot's clear. What? Did you wipe it clean it? Yeah, good. All right, good. Because I just don't want anybody sticking their hands in anybody's spit. All right, let's go ahead and we'll start. If you're still in the kitchen working, that's fine. Just work quietly and try to minimize mess as best as you can. So we are doing our little last bit on DNA, and I'm actually wearing it again. Yes. All right, so let's get started. Nucleic acids. So nucleic acids, this is a model of a single chain or just one part of a DNA helix, for example. And what are they composed of? A chain made of what along the backbone? What do we call the chain? Caitlin?
RNA.
RNA or DNA are types of nucleic acids. This chain right here with the P's and the pink things. Does anybody know what that is? What you got? Caitlin? Do it? Nucleotides, gonna be those. Do you know?
That's ATP.
Not ATP. All right, so what we have is a phosphate sugar chain. So if you look right here, you see that nice diagonal chain made of the little pink pentagons and the little P's? What do you suppose the pink pentagons are representing? Sugars, and the P's are? Phosphate. So you have sugar, phosphate, sugar, phosphate, sugar, phosphate, and all of the sugars is where you find your nucleotide bases, right? So our G's, T's, A's, C's, U's, depending on which nucleic acid we are talking about. That's Jack's chair, yep. So, how many bases form a double helix for DNA? How many bases, Colin? Overall. Two linked together, how many are there in total that can be used?
A lot.
A lot? No, not a lot. How many, PJ? Four. He's like, now I know what you're asking me. Yes. All right. 4. He's like, I don't know what you're asking me, lady. 4. Okay. So you have 4 bases. And in DNA, A goes with what? T, and G goes with C. Now, if you look, we have of these different, the different nucleic acids, we have DNA and RNA. DNA is deoxyribonucleic acid, and RNA is just ribonucleic acid. So when you look at these, you'll see that DNA only has thymine and deoxyribose, thus it is DNA. And remember that in RNA, you don't have thymine, you have what? Uracil. There's the U. Remember last week when all of your T's had to be transcribed into U's?
Yeah.
Okay, that's why, because we were transcribing 2 messenger RNA. So when you do that, you need to have U's instead of T. So in your RNA, you have ribose and uracil, but in both, you have adenine, guanine, and cytosine. Those are your A and your G and your C. And then you have your phosphate. So based on knowing that it's a phosphate sugar chain, what type of molecule would deoxyribose and ribose be?
Sugar.
Very good, sugar, because you have already your phosphates, and the sugars are what your nucleic acids attach to. And if you look here, they've shown them like puzzle pieces that fit together. And although that shape itself is not really accurate, the concept is you're talking about shapes that link and fit like puzzle pieces. That's why those particular molecules will bond. They won't bond with the wrong pair. So A will not bond with G because it's not, the shape isn't correct and the bonding sites are not correct. All right, so if you look at your base pairing where we talked about C, G, T, A, right? We actually call those different things. Does anybody know which 2 are going to be called pyrimidines? So your pyrimidines are going to be your thymine and your cytosine, and then your purines are going to be your adenine and your guanine. So when you always have a pyrimidine and a purine together, right? Because A and T right there go together, C and G go together. 1 pyrimidine, 1 purine. And if you look, when I said a minute ago that the binding sites are only going to work with the correct pair, you can see right there why. There's your pyrimidines your pyrimidine and your purine there. So your thymine and your adenine, those bonding sites allow those 2 to attach to each other. And your cytosine and your guanine, their binding sites allow them to attach to each other. But you're not going to be able to put guanine with thymine or adenine with cytosine because it's not going to work. The bonding sites are not going to be the same. And if you look at this graphic here, this shows you how you get messenger RNA coming in or replicating. And what you get here, this little guy, we'll learn a lot about him next year, but that little blue thing is called helicase, and it essentially unzips the DNA, just like your zipper pull will unzip a zipper on a hoodie. It's the exact same thing. It will unzip it, open it up. And I have a good video that'll show you how that happens in a minute. So the arrangement of base what on DNA? How do they work? Is it 3 at a time, 2 at a time, 1 at a time? What did you do last week in lab? 3 at a time, right? So it's triplets. And what's the other word for those? What do we call those? Codons. Very good. A set of 3 are called codons. And we have a start codon, a stop codon, and everything in between. What does a codon on your DNA tell your messenger RNA to make? What does it do? What it binds to it? Proteins. But first, that one codon is going to give you, that codon tells it which amino acid needs to be there, right? And then codon after codon after codon puts the amino acids together in a chain, and the chain makes your proteins, right? So incidentally, it is not uncommon for there to be miscoding, and we call those mutations. And you probably have many, many of them in your DNA, and many of them are not harmless at all. Some are extremely harmful and some are deadly, depending on which, where the problem is. Yeah,
We can't become Godzilla.
not Godzilla.
Is cancer also something like
It
that?
can be. So that is a kind of a down the road result. But if your DNA gets damaged, say by UV light, okay, like you're out in the sun and the sun hits your skin and you get sunburned, it can damage the DNA. And if it damages the part of the DNA that tells your cell how and when to divide and how frequently and when to die, then yes, that can give you cancer. Because cancer is like the uncontrolled constant division. For example, if you have, say you have kidney cancer, when it metastasizes, if it fills your whole body and they find it in your brain, those are kidney cells. Like that's what you're getting. It stays within those cells that continue to replicate uncontrollably. So yes, that can cause cancer, damage to your DNA. They call things the damaged DNA tetragenic, fun word. So this is what it looks like when it replicates and is transcribed. Actually, this is more transcription than replication, but I got to turn this volume up. Get it down real low. The music earlier. Okay, now we can hear it.
It's a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of 3 bases in the nucleus.
And that's what you did in lab last week,
The
right?
DNA code determines the order in which the 3 bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acids. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
So that's happening in your body all the time, constantly, very quickly, very, very quickly. And that's how you get, that's basically what we did last week, right? We created proteins. In our case, our analogy was a sentence. All right, so moving on to the types of technology we can do with DNA. So back in the late 80s, early 90s, we started using DNA fingerprinting and profiling. So this is your example here. If you look right here, this would be DNA collected in an investigation, for example. So you have your victim, 2 people you suspect might be involved, and then you have DNA collected from your crime scene. So whose DNA is that where it says crime scene 2? Can anybody tell? Can you tell, Lucas?
Victim.
It's the victim. Do you see how it matches? And then who did it?
Suspect 2.
Suspect 2. We all see suspect 2 did it. Does everybody see why suspect 2 did it? Can everybody tell? You can't, yeah.
It matches the one at the crime scene that did.
Yeah, it's the same pattern. Now, way this is done is actually really cool. And we will do this next year in lab. We will do this exact same thing. What we will do is we will take some DNA sample and we will put it into electrophoresis gel. That's what this is. So electrophoresis gel is really cool. It takes the DNA and it runs an electrical current through it and it breaks it down into these markers that make these little lines. And it will be that same color blue and you'll look at it on a light and you can see exactly where it is. And we will do this exact thing and basically solve a crime next year in lab. That's one we've done before. That's not the same one that we're going to be doing. That's the multi-week one. That one's different. But this we'll do on just one day. Now skip the one in the middle and go to this guy over here on your far right. So we can use recombinant DNA, which is another thing that we do. And it can be for vaccines, insulin, growth hormones, pest-resistant plants. Basically, we can take the genes we want, we can plug it into bacterial DNA, for example, and let the bacteria go ahead and do the replicating for us. And then we can take that back out and plug it back into whatever organism we want to plug it into. So that's yet another way. Now this one in the middle, cloning. All right, we're gonna watch a video on Dolly the sheep. Anybody heard of Dolly the sheep before? Oh yes, okay. So this happened in 1996. So I was right in the middle of my biology degree when this happened. So it was big news. Like we were all over it. Everybody was all over it. And actually there's a the student newspaper at Hollins is called the Hollins Columns. Haha, because there's columns on the front porch, and Hollins Columns, and it's kind of rhymey. They had an April 1st edition, and that's always an April Fool's edition, and it's always goofy and makes no sense. And one of my friends, she's now a physician, she's one of my biology major buddies, she wrote an article for it where she was the clone and I was the surrogate. Like I carried her clone and then delivered her clone. It was this whole thing. It was great. It was a great article. So anyway, I carried a clone in a fake article once. But basically what they did is they took a somatic cell meaning it is a body cell, which is diploid, meaning it has 2 copies of the DNA in it, like all of ours do, not haploid, those would be gametes, right? Have half your DNA. So it took a diploid somatic cell, in this case, a mammary cell, from one sheep, and pulled the DNA from it, took an egg from another sheep, removed its nucleus, took the DNA and all that information from the mammary cell, injected it into another, basically an empty egg cell, popped it into a third sheep, and then that third sheep gestated the baby and birthed the lamb, and that lamb was a clone of the one who they borrowed the mammary cell from. She was the first mammal to be successfully cloned. Lots of Scottish people carrying on about how awesome it was. So yeah, so this is Dolly. Well, that's actually the stuffed version of Dolly that's now in a museum. She's taxidermied, and that's the one they showed the picture of there. So you get taxidermy Dolly in your picture there. Okay, yes.
Dolly the sheep was a sensation when she first appeared before the world. A scientific breakthrough, fascinating, but also menacing. Dolly was cloned by taking a body cell from sheep A, fusing that cell's DNA with an egg cell with its nucleus removed from sheep B. This fused cell developed into an embryo, which was placed in the uterus of a surrogate mother, sheep C. The resulting lamb, in this case Dolly, is a clone of sheep A. These were Dolly's so-called siblings, identical sisters, all cloned from the same sheep. Professor Kevin Sinclair worked with them and knew Dolly.
It was a step change in our understanding of early development, which ultimately led several years later, in 2006, to the discovery that you can actually induce somatic cells. These are cells that belong to the body, to become embryonic-like cells that has the potential to divide into other cell types.
This was Dolly's greatest legacy, advances in what we call stem cell research. The ultimate, but still elusive goal is to use cloning technology to create healthy tissues which can be transplanted to heal damaged cells and organs. But back in 1997, Dolly seemed to raise more frightening questions. If we can clone a sheep, many asked, why not a human being? Since then, scientists have cloned more than 20 species, including horses and household pets. But scientists have also recoiled from using these technologies on humans.
I think where we draw the line is if we're going to use genetic manipulations, we have to be extremely careful, and we have to look for severe disease effects to control for. Because with a lot of these things, it's not just the person you're doing a genetic manipulation on or potentially cloning, but it would affect all the generations ahead.
So Dolly's brave new world has not quite turned out as predicted. A sheep created like none before her, but she could not escape her own mortality. She died in 2003, now stuffed in a Scottish museum.
I love the way
Now
now stuffed in a Scottish museum. So
Didn't she die young or something like that?
a little, I think, yeah.
So that's what I wonder. I feel like I hear that a lot where cloned animals die young, but if they're genetically identical, why are they dying young?
It's a good question, isn't it? That's where we need our additional research. Based on what we know and what we understand in general, that shouldn't be an issue, but we're starting to understand more. Incidentally, a couple years ago, there was a whole article about how they had cloned meat that cloned beef and they made a meatballs. They were making these little meatballs and people were eating the meatballs and like, come eat the meatball. How does it taste? Oh, it tastes like meat. People were panicking like, I'm not eating that. That's clone meat. Theoretically, based on what we know and what we understand, your body shouldn't be able to tell the difference between cloned beef and beef that came from a cow that's, these were like cloning it, growing them in a petri dish and making meat, right? Like beef muscle cells, right? So technically, theoretically, your body should know the difference. Does it? Yeah.
So Dolly died early because she was called.
I think she died a little early. The question is, we don't really understand. I'm not sure what the lifespan of a sheep is. Yeah.
How old was sheep A?
I don't know.
Because
It's a good question.
If you're an adult sheep and she has the DNA of an adult sheep, then she's already
Mhm.
completely close to dying. The adult sheep has the same DNA she did when she was a baby.
Yeah, the DNA. So when it comes to that, so that's an interesting point. Like, is taking older DNA, start that organism off older? It shouldn't, because you're older because all of the processes in your body have gone on over a certain number of years. But embryologically, you started with the same DNA. So embryologically, Dolly started with the same DNA as her clone mother did, embryologically, and then developed from there. So the timeline should be the same, but it's not necessarily.
Dolly died when she was 9, sheep like 10 to 12 years.
Okay, so a little early. A ton. And then, it's like, this is something that's really cutting edge and we don't know a ton about it. We're still working on it. We know way more than we did 25 years ago when I was just learning about it. Now it's, there's a lot more 25, 30 years ago. Now, another thing to think of, they mentioned stem cells. What are stem cells? Anybody know what a stem cell is? Non-specified, non-differentiated. So stem cells, you can find stem cells in your bone marrow. There are a lot of them in embryos, but they are basically, it's a cell that hasn't decided what kind of cell it wants to be yet. Its DNA is going to tell it whether it becomes skin, bone, brain. Yes.
Babies have a certain form of stem cells.
Yes.
Teeth.
It'll have to be very embryologically, probably not too much older, but
There's also stem
Hang on.
cells in our nail bed.
Yes, in your nail bed. Yep, guys in the back.
Let's grow it back.
Yeah, the very tip, not the whole thing, but tip. Yeah, like he was saying, because there are some stem cells in your nail bed. So yes, so the idea would be, if we can take, let's say something terrible happens to James and he needs a new liver. Okay. Oh no. The idea would be, if we could take stem cells from James that are genetically his and engineer them to differentiate into liver cells, we could grow James a whole new liver. And we could take out the bad liver and pop the new liver in and boom, there's no organ rejection because it's his DNA. That's the idea.
How long would it take to do that? Longer than you would have.
We don't know. They haven't been like really successful. Now they have 3D printed some organs using certain types of cells, which is very fascinating to me. But
Yeah.
yes, ma'am, Ridley.
There's also a way to have my kids, the same.
Yes. So we're gonna get into that in a minute. So it's not really like a legal, it becomes questionable. So
Get sick, I have another one.
yes. Well, some people, there are some people who in the past, this is where we get into a serious ethics discussion and it gets really interesting. But there are people who will have if they have a child with a disease that's going to require them to have lots of blood transfusions or things like that, they will have a second child as basically like, it's happened a few times. This is like our farm kid. We're using them as a source. Yeah, there have been people who have done it. It's not common, but it becomes kind of it's a very questionable practice, obviously. Do you have a second child to be a source for your first child? So a stem cell has not yet differentiated, and DNA within that cell is going to tell it what to become. Could become skin, bone, liver, whatever, right? And stem cell research is fascinating and complex, but it's not without potential harm. So I said, you can get stem cells from bone marrow and places in your own body, but a really good source for stem cells are human embryos. And here's where it gets sticky, ethically speaking. So in the US, stem cell research, the federal government doesn't ban it, but it limits the funding. By limiting the funding, you control a little bit how the research is going because it's very expensive. That doesn't mean you can't get private funding. You can. Okay, so we've limited some funding. The NIH, National Institutes of Health, prohibits federal funding for deriving stem cells from human embryos. Okay, fertility clinics. I don't know if you all saw it, there was an article just this last week that this woman had delivered a baby that she got implanted as a donated embryo. And I believe the woman was born in 1991 and the embryo was created in 1992 and has been in suspended animation and cryo freeze all this time. And they successfully implanted it and she delivered a baby that's really like, you talk about when it was conceived, when that DNA was formed, when that individual human was formed, a year after she was born or something to that effect. Wild, right? Really wild. But not born, but began developing. So that's interesting, right? So if you go to a fertility clinic, and a lot of people do use fertility clinics to help have babies. So, okay, you need help having a baby. They harvest eggs. They harvest a lot of eggs. Typically, you only get one of those a month, but they pump you full of hormones, so you produce a lot. They'll harvest a lot of eggs. They'll harvest the sperm. They will go ahead and fertilize all the eggs. Then they will let them start forming into embryos. And those that start forming, they will freeze the ones you don't need. Now, they're not typically going to go implanting just one egg because it doesn't always take. That's why people that go through fertility will often have multiples because they'll put in 2 or 3. Now, at the beginning of this whole thing, they used to throw in a lot because they weren't sure how many would actually implant and start developing. So there was this lady, you can probably look her up. Does anybody know who I'm talking about?
Yeah, Octomom.
Octomom. Everybody knows Octomom? Octomom had a whole ton of embryos implanted and they all took, or most of them took, and she was pregnant with 8 babies. That is not something that humans are developmentally meant to do. It's not healthy for her. It's not healthy for them. So we've done enough. Yeah, we've done enough. Oh, the pictures of her pregnant are ridiculous. She was huge. Obviously, babies are going to come early because there's no room. But here's the thing. We've gotten to the point where we've done this for long enough that we have an idea of kind of the safe number to implant, where you're likely to get some to take, some will not take, they will die. But usually you have leftovers and those remain frozen. Now you can keep them frozen and say those are my embryos and I'm going to keep them frozen. You can donate them and some of them just sit. So then the question becomes, well, if no one wants these, do we use them for research? And that's where it gets sticky because some people will be like, yeah, and other people will be like, no, absolutely not. Question.
Did Octomom have to get a C-section?
Oh yeah. Oh yeah.
No.
Yeah. Oh yeah. And not because it's not physically possible to vaginally deliver 8 babies, but because they needed to come early because there was no more room and they were going to be fragile and needed to be like immediately reversed. Yeah. You can safely vaginally deliver triplets and whatnot. I mean, I know a lady who had a VBAC after 2 C-sections. So she had 2 C-sections and then she had a VBAC, which is a vaginal birth after C-section. So her 3rd birth though, her VBAC was triplets. So she had 2 C-sections with singletons and then vaginally birth triplets and she did it with my OBGYN. They were great. They did a great job. So yes.
On like at the very beginning of one video, there's this giant baby that's only like 1 year old.
Oh,
But it's like
probably has some hormone issues. Yep. So we get into this issue of what do you do with embryos? And it's say they're your embryos and you want to donate them to science, and that's your choice. Some people think it should be, some people think it shouldn't be. It gets very, very, very sticky. So however, research that doesn't destroy embryos is federally permitted. So if you're not destroying an embryo, if it's miscarried or whatever, and you're able to harvest it, which sometimes that will happen, it's already deceased or whatever, then they will go ahead and use that. But that's different than destroying embryos that are in cryo. Like it's a different situation. All right, and then state laws are going to vary widely. Some states support stem cell research, others completely ban it. I don't know where Georgia stands. Scientists often use these leftover embryos from fertility clinics, right? Now, cloning. What are our rules on cloning? This is constantly changing, by the way. So the government doesn't prohibit cloning, but it does prohibit the use of federal funds. Some people are staunchly against it for religious reasons or personal reasons. They're like, no, you shouldn't be cloning human cells, period. And where does the government get its money? From taxes, right? So that means if you disagree with something and they're paying for it, they're using your money to pay for something you disagree with. That's why the government has decided this is something that they're not going to federally fund. Privately funded? Sure. Right. Now, this applies to both therapeutic and reproductive cloning. Yep. Like, let's try and replace these damaged cells or cancer cells with healthy cells, that kind of thing. Policy changes. There's a great policy name in here. You know how sometimes they're named after people? Yeah. There's a real good one in here. We're getting to it. So in 2009, Obama overturned Bush's policy, which limited federal funding. So now there's like a little more for stem cell research, still not cloning. I love this one. The Dickey-Wicker Amendment. The Dickey-Wicker, it's just fun to say it, Dickey-Wicker. The Dickey-Wicker Amendment prohibits destruction of embryos to create human embryonic stem cell lines. Okay, that's like a whole thing where you get these lines of embryonic stem cells that you can use for different applications. Yeah. Yes, there is. It's a really, it's highly inappropriate, but it's hilarious. It featured a cartoon version of Christopher Reeve, God rest his soul. So this is initially one of the promising things about embryonic stem cell research was using for spinal injuries. So you can regenerate spinal cells, nerve cells, and spinal injuries. You can potentially get somebody who's been, an accident caused them to be paralyzed. Maybe you could restore their, mobility. That's the idea.
Dickey-Wicker Amendment?
Mm-hmm.
Is that like an amendment of the Constitution, or is that, what is it amending?
I think it amends a previous law, a specific law. Mm-hmm. But I'm not sure. So energy in living systems. So what do we notice about these two equations? Photosynthesis and aerobic respiration are what, Jordan?
They involve each other.
They are opposites of one another. Yep. What are the reactants in photosynthesis?
Glucose.
In photosynthesis, the reactants are carbon dioxide and
Water.
what are the products in cellular respiration? No products. Carbon dioxide. Y'all know reactants and products. What are the products in photosynthesis? Glucose. That's glucose and oxygen, and then your reactants in respiration are going to be. Photosynthesis are going to be respiration. I'm back. Yeah, sugar, right? Glucose and oxygen. So green plants use solar energy, carbon dioxide, and water, and what do they synthesize? What are they making? Right here. C6H12O6, glucose. There you go. During photosynthesis, the reverse process is cellular respiration, in which glucose is broken down into, what do we break it into?
Water.
Carbon dioxide and water. Energy is harvested in the process by the production of 38 ATP ions per glucose molecule. There's this whole dealio called the Krebs cycle, which you will learn in biology. But this is basically what happens. Through the release of energy during breaking of its third phosphate bond, ATP fuels life's processes, motion, synthesis, transportation. You are making millions and billions of ATP molecules over the course of a day. It's just what's happening within your body all the time. Every single glucose molecule is going to yield 38 of them. And that's what we use for our energy. This occurs in the mitochondria. If you've heard the mitochondria called the powerhouse of the cell or the power plant of the cell, that's why. It's where we're taking the products of cellular respiration and converting them into energy we can use. So here's our amoeba sisters again, who are always so fun.
Why wait?
Go to med school.
And to get hands-on experience within.
You'll make money. Pre-med's not that. Yeah. If I can do pre-med, you can do pre-med.
No. And one of us is definitely not. Mainly because when I wake up in the morning, it just takes a while for me to get my energy back.
It takes a lot of time and coffee for that to happen for me. Cells really don't have that luxury. They are busy performing cell processes all the time. Active transport of many substances needed for their survival. And the energy currency they need, specifically, is ATP. ATP stands for adenosine triphosphate. It's a type of nucleic acid, actually, and it is action-packed with 3 phosphates. We have a video all about ATP and how it works as an energy currency. So where am I going with this? Well, cells have to make this ATP. It doesn't really matter what kind of cell you are, prokaryote or eukaryote, you have to make ATP. The process for making that ATP can be different, however, depending on that type of cell. One way is called aerobic cellular respiration. Lots of organisms can do aerobic cellular respiration, but this video is specifically going to go into aerobic cellular respiration in eukaryotic cells. That means this video is talking about the process within cells that have membrane-bound organelles, such as a nucleus and mitochondria. Eukaryotic cells include the cells you find in protists, fungi, animals, and plants. The mitochondria, which can be found in most eukaryotic cells, are going to be kind of a big deal in this aerobic cellular respiration because some of the process occurs in the mitochondria. So let's get started. Remember, the major goal for any organism performing this is to make ATP. Okay, here's the overall look at the equation for aerobic cellular respiration. Remember that reactants, inputs, are on the left side of the arrow, and products, outputs, are on the right side of the arrow. This equation, by the way, is remarkably similar to photosynthesis. Look at how the reactants and products are on different sides. Well, that doesn't mean they're simply opposites. It does show that they have substances in common. In photosynthesis, organisms make glucose. Notice how glucose is a product. But in cellular respiration, organisms break the glucose down to make ATP. Fun fact, you know how when a bean seed first germinates in the ground, it isn't able to do photosynthesis yet? Yeah, the germinating bean is relying on glucose that it has stored, and it's doing cellular respiration to break it down, make ATP so it can grow. Of course, once it starts to mature and develop leaves, it can do photosynthesis. It will be able to do both photosynthesis and cellular respiration. Plants make glucose in photosynthesis, and they break it down in cellular respiration. They can do both. But if you aren't photosynthetic, such as a human or an amoeba, you have to find a food source to get your glucose. You need glucose to get this process started, and we're going to assume that we're starting with one glucose molecule. Step 1, glycolysis. This step takes place in the cytoplasm, and this step does not require oxygen. It's considered anaerobic. In glycolysis, glucose, the sugar from the equation, is converted into a more usable form called pyruvate. Glycolysis usually takes a little ATP itself to start up. The net yield from this step is approximately 2 pyruvate and 2 ATP molecules. And 2 NADH. What is NADH? NADH is a coenzyme, and it has the ability to transfer electrons, which will be very useful in making even more ATP later on. We'll get to that in a minute. Now, an intermediate step occurs. The 2 pyruvate are transported by active transport into the mitochondria, specifically the mitochondrial matrix. In the mitochondria, pyruvate is oxidized. The 2 pyruvate are converted to 2-acetyl-CoA, which will be used by the next step. Carbon dioxide is released and 2 NADH are produced. Step 2, the Krebs cycle, also called the citric acid cycle, still in the mitochondrial matrix. The citric acid cycle is considered an aerobic process. While the cycle doesn't directly consume oxygen, some of the events in the cycle need oxygen to continue. To learn more about this intricate cycle where the 2-acetyl-CoA will enter, check out the further reading suggestions in the video details. Carbon dioxide is released. We also produce 2 ATP, 6 NADH, and 2 FADH. FADH is also a coenzyme, like NADH, and it will also assist in transferring electrons to make even more ATP. Step 3, the electron transport chain in chemiosmosis. This is just a beautiful thing, really. In eukaryotic cells, this is still in the mitochondria, and to be more specific, it's involving the inner mitochondrial membrane. We do require oxygen for this aerobic step. This is a very complex process, and we are greatly simplifying it by saying that electrons are transferred from the NADH and FADH to protein complexes in electron carriers. The electrons are used to generate a proton gradient, as protons are pumped across to the intermembrane space. All these protons being pumped out into this intermembrane space generates an electrical and chemical gradient. The thing is, if you remember from our cell transport video, ions, like the H plus, don't easily travel across membranes directly without something to travel through. The protons can travel through an amazing enzyme called ATP synthase. If I could be any enzyme, I'd be ATP synthase because it has the ability to make ATP by adding the phosphate to ADP. ADP is a precursor to ATP. ADP has two phosphates, but if it obtains a third phosphate, it becomes ATP. So in chemiosmosis, the protons travel down their electrochemical gradient through a portion of the ATP synthase, powering it to make ATP, the ultimate goal. Oxygen is the final acceptor of the electrons. When oxygen combines with two hydrogens, you get H2O, water. If you remember from our equation, water is a Listic product. Now, the electron transport chain in chemiosmosis step makes a lot more ATP compared to the other two previous steps. How much? So I've noticed in my years of teaching and the assortment of textbooks I've collected over the years, there are some different numbers in cellular respiration charts. In fact, you can even see them change a bit in different editions of the same book. And it's made me want to emphasize that it's important to not just memorize a number, because it really is more of a range. I want to focus less on the number of ATP made per glucose molecule because it depends on a lot of variables. One variable is the gradient we were talking about. How many protons were pumped across the mitochondrial membrane? You can see more variables discussed in the factual references. And those references have estimates ranging from 26 to 34 molecules of ATP per glucose molecule in the electron transport chain in chemiosmosis step alone. Then if you add the other two steps, Krebs, otherwise known as the citric acid cycle, and glycolysis, you can estimate a range of anywhere between 30 to 38 net ATP molecules total per molecule of glucose. Again, to emphasize a range. Now, this is just one way of creating ATP. But like we had said at the beginning, all cells have to make ATP, but the way they do it can differ. If there's no oxygen available, some cells have the ability to perform a process known as fermentation. It's not efficient, but it can still make ATP when there isn't oxygen. We have a video about that. We can't emphasize enough how important the process of making ATP is for cells. For example, cyanide, which is found in some rat poisons, can block a step in the electron transport chain, which would block ATP production. A poison that prevents ATP from being produced can be deadly. With the important role that mitochondria have in ATP production, there is also a demand for increased research on mitochondrial diseases. We are confident that the understanding of how to treat these diseases will continue to improve as more people like you ask questions. Well, that's it for.
So ATP, we'll talk more about it next year, but that's a little jump ahead to give you an idea of what we're looking at. So that's the goal of aerobic respiration, is to produce ATP, and that's where you often see it just listed as like energy. That's what they're talking about. All right, let's talk about viruses now. All right, viruses have DNA. How do viruses replicate? Like so. So keep this in mind.
They all have a similar structure, being surrounded by a protein shell called a capsid, which protects the viral genome. Inside this capsid is either viral DNA or viral RNA. Also, is the viral protein, which is covered in attachment proteins. The viral envelope is made of lipoproteins. So how do viruses reproduce? They don't quite lay eggs, that's for sure. Viruses don't have a way to reproduce alone. Instead, they need to hijack a host cell. To do this, they approach a cell. That attachment protein we mentioned earlier interact with specific receptors on the cell membrane. The protein and the receptor are complementary. The contents of the virus, such as viral RNA, the reverse transcriptase enzyme, are then injected into the cell by endocytosis. The reverse transcriptase enzyme copies the viral RNA, forming double-stranded DNA, which is then transported to the nucleus. The viral DNA is integrated into the host DNA, and the new host cell assembles new viral DNA and the proteins necessary for the capsid and envelope. The new viral DNA and enzyme then leave the cell by exocytosis, constructing a viral envelope in the cell membrane of the cell. Eventually, this will kill the cell, while the virus will therefore infect other cells, and the whole cycle begins again.
So viruses need to hijack a cell's DNA, RNA production replication in order to make more of themselves. But here's the thing about viruses. Can you kill a virus? Who says yes, you can kill a virus?
People?
Who says who says no, you can't kill a virus? Okay, you can kill a cell. How about this? Can you kill a bacteria?
Yes.
Yeah. Define kill something.
Unalive body.
To unalive it. So to kill something, it has to be alive first, right? So then the question becomes, are viruses alive? Not really. No. Uh-huh.
Can we call killing it, making it cease to function?
Hold on, I can't hear Caden. Say it again.
Can we make, is killing it considered making it ceasing to function?
Maybe. So here's the thing. You can take, if you have strep throat, what do they give you?
Antibiotics.
Yeah, they give you an antibiotic. It will actually kill the bacteria in your body. If you have a flu or something like that that's caused by a virus, they might give you an antiviral. It doesn't kill the virus, it inhibits viral replication. But once you've had the virus, you've got the virus, right? It hangs out. This is why, like, you don't catch the same exact cold again, because now your antibodies have decided they've recognized it. It's a whole production thing. But the viruses aren't really alive to kill in the first place. So viruses use a host cell to replicate and reproduce. They put their RNA in, they have single-strand RNA, and then they allow the host cell to do the work to produce more of their single-strand RNA. So we come into contact with viruses every day. The common cold is a virus. You haven't had the same common cold twice. Actually, most common colds are coronaviruses, just not the particular one that shut everything down a few years back. So you can treat symptoms, but since it isn't really alive, you can't take a medication to kill it like you can if you have a bacterial infection. Antiviral medications inhibit replication. What you can do, however, if you have, say you have a virus that some viruses can live on surfaces for a certain amount of time, right? If you clean that surface with particular cleaning agents, alcohol being a good example, you can denature the protein membrane that holds that viral messenger RNA, and then you've basically disabled it.
They can
Live. Yeah, exist. How about that? They can exist. Thanks for correcting me. They can exist on a surface. They can be there and not, y'all all right? Quit touching her. Stop touching. Hands are for hugging. Anyway, Drew's like, man. There you go. So if you have strep, you can take an antibiotic and you'll be better soon because the antibiotic will kill the bacteria that makes you sick. But if you get the flu, for example, you can treat your symptoms while your immune system does its work. And you can take something like Tamiflu, for example, or Paxlovid, which they came up with for COVID. And though you can't really, it'll help prevent it from replicating, but you can't really kill it because it's not alive. Do you remember what I said about Paxlovid being interesting? Because it was an isomer of ivermectin, right? So yeah. All right, so some viruses are bothersome. Common colds are bothersome, generally. Nobody likes the common cold, right? Nobody likes getting the flu. That's really bothersome. Some of them are deadly. Some viruses will kill you. HIV, which we've now got largely controlled, and we're about to see why, or very largely controlled compared to how it started. It's human immunodeficiency virus, and if you get HIV, it over time can develop into AIDS, which is acquired, meaning you weren't born with it, you acquired an immunodeficiency syndrome. Some people just have an immunodeficiency syndrome that they were born with, it's congenital. In this case, though, it is acquired. You catch a virus that attacks your immune system to the point where you're now immunodeficient, meaning your immune system doesn't do what it's supposed to do. And while you can catch some viruses by inhaling somebody's cough, HIV comes through sexual contact, sharing needles, mother to fetus, breast milk, or blood transfusion. Blood transfusion really rare these days, obviously. So you don't get HIV from using food or utensils that somebody who is infected has. You don't get it from insect bites. It's not one of those viruses that can be transported, for example, by mosquitoes. Some things can. Some viruses can. You don't get it through kissing or touching. You don't get it through clothes or towels or from a public toilet or a shower, but you do get it from use of non-sterile syringes or tools. This is mainly affects drug addicts that are sharing needles because this doesn't happen in doctors' offices. You've seen them. They give you a shot or they draw your blood. They immediately put it in the sharps container, right? Pregnancy and breastfeeding, blood transfusion, organ transplants, and unprotected sex. So which two do you suppose are our most common?
Sex.
Unprotected sex and what's the other?
Needles.
Sharing needles. These are the ones that'll get you. Pregnancy and breastfeeding, for the most part now, people know they have HIV before they get pregnant and they don't get pregnant, or they can be treated during the pregnancy to help make it so that it doesn't pass. And then those people don't breastfeed because they don't want to pass the virus through breast milk to their baby. Blood transfusion, we test everybody now. When they donate blood, they test you for HIV. So you're not going to get it through blood transfusion, at least not in the US. You'd have to be somewhere real weird for that to happen. Organ transplants are the same thing. Donors are thoroughly tested for anything that could be passed on to a recipient. So you're not going to get it that way. Now, this is one of the drugs that's been recently developed and is making a world of difference for people that do have HIV. Woo, what is this? This is fun. I don't know what that is.
Years since Benta Golan first found out she was HIV positive. At the time, she was only 27 years and 6 months heavy with her fourth born child. But it has been a long walk to living freely but positively due to the stigma associated with the disease.
We were pointed out, we were talked about, people still talked about us, and it was sad. For some time, I told my cousin I don't have to go for medication.
A situation that a majority of the 1.5 million persons in Kenya living with HIV have experienced at one point in their lives, but a situation that could change given the latest approval by the US Food and Drug Administration of a two-shot combo called Cabenuva. This becomes the latest innovation in treating what has become a chronic, manageable, but still incurable disease. An innovation that gives hope of reaching groups that have found a hard time sticking to treatment, people with mental illness, and those that have substance abuse problems.
It's going to make it easy for people to actually take their medication. And hopefully, I think it's going to improve the quality of life for people living with HIV.
That would be good because I'll organize myself. I know I'm not going to go for injection then I come back. Yeah, so people will not be hiding themselves that she's just been tested. She's not telling anyone. She's not going for medication. People will come out.
The injectable known as Cabenuva is a combination of two drug formulations, cabotegravir, which is a new drug, and also one described to be able to prevent one from being infected with HIV, and rilpivirine, a drug that has been used before in the treatment of HIV-positive persons. The drug packaged together will be given a separate shot once a month.
And we've come from far because we came from a time when people take up to 18 pills a day. And there will be challenges because they have to take some of these pills with food. So there are challenges getting food.
I think I'm so much exhausted. Before I watch 9 p.m. news, I'm already asleep. Sometimes I forget. So it has been a hurdle.
According to the FDA press statement, an approval has also been made for Vocabria, which is the cabotegravir tablet formulation, and which should be taken in combination with oral rilpivirine for 1 month prior to starting treatment with Cabenuva. This is to ensure the medications are well tolerated before switching to the extended release injectable formulation. For Dr. Peter Cherotich, a public health and HIV/AIDS specialist, the cost of the medication will be expensive in its initial stages, just like other innovations. However, given the commitment of donor funding, the drug will be made affordable over time.
First, for you to get your injectables, your viral load must be well controlled. Of course, there's still challenges on children and dosing, but that will come later on.
At the moment, a majority of the persons living with HIV are taking 1 pill daily, which is a combination of 3 other drugs. And while the injectable will replace the daily pills,
A HIV positive person will be required to take other medications that will prevent them from opportunistic diseases. Rakami Guy, Citizen TV.
So they used to treat it by up to 18 pills a day. So think about compliance, patient compliance at taking 18 pills a day. That's hard. It's hard to get your patient population to comply with 18 pills a day, every day for the rest of their lives. And then they narrowed it down to a pill a day, whatever. Still, anybody who takes daily meds knows sometimes you forget. Maybe you have to take it with food. Not all of these people have access to food all throughout the day. So it gets to be sticky and tricky. So now they have, you go once a month, you get shots, and you're good for a month because it's extended release. Notice they said they have an oral version that you take for a month prior to getting your first shot. That's because if you don't tolerate it, you can stop taking the oral version. You can stop the pill. If you get the shot and you have a bad reaction, you're screwed. Because now you've got the shot, right? There's no way to back off and go, well, I'm not going to do that anymore. You already have it. So we are in a way different place than we were in the late 80s, early 90s. I mean, we've come a long way with HIV treatment, and thank goodness, because it's awful. So now we're going to take a few minutes and we're going to do a little research. I'm going to give you about 5 minutes to look up some things here. DNA phenotyping. So this is cutting edge forensic science. So I want you to take a couple minutes and research what is DNA phenotyping. What role has a company called Parabon Nanolabs played in cases of unidentified remains, unsolved murders, or cold cases? Take a few minutes and see what you can find. I'll give you like maybe 4 or 5 minutes. You don't need too long. It's 1:03. But 2 more minutes. Okay, who'd like to share something they found? Jordan.
Parabon Nanolabs
Mhm.
is a company that provides the DNA phenotyping to law enforcement to figure stuff out.
Very
And
good.
the phenotyping is trying to reconstruct something from a DNA source.
Very good. Very good. James?
They use technology, I forget what it's called, Snapshot.
Mm-hmm.
They use Snapshot, which is a
Yep.
program they made to take the DNA sequence from unidentified remains. And from that, they can tell the phenotype, which is what a person looks like.
Right, right. Anybody have anything else interesting they found? Caitlin, what'd you find?
Parabon Nanolabs develops non-pharmaceuticals and provides a DNA phenotype.
Yes, okay. So a genotype versus a phenotype. A genotype is what your genetic code says. A phenotype is what you present like from your DNA code. There is a gene that says brown eyes. That is a genotype. The actual brown produced is the phenotype. So what Parabon has done, they were on the cutting edge of this, and now they're not the only ones that do this type of thing. But what Parabon does for their snapshot, their phenotype snapshot DNA, is they will take DNA from either unidentified remains or from an unknown suspect, they'll call them an unknown subject, and they will take that genetic code and then create an image of what that person looked like. So in this case, this young lady's remains were found. And if you notice, it says like female, age unknown, body mass unknown. That probably means you're dealing with what kind of remains? Yeah, you don't have a full body to deal with, you're dealing with skeletal remains if they can't estimate body mass, right? They can give a rough estimate, but not real good. And then they took all of her genetic code and spat out that image as her phenotype. Now they say, this is your person. This is who you're looking for. This is our person. Anybody know this person? And some agency somewhere that has a missing person that looks like that goes, oh my gosh, we're missing this young lady. I believe Shaquan Caldwell was her name. There she was. Oh, we're missing someone. We're missing her. So then all they do now is they take the DNA from her that they got from her remains and compare it to a family member and confirm it's her. In doing so, now that they know who's mitten, now they know this person didn't just leave. Full-grown adult can just leave. They can walk away from their life and just leave, right? Now we know she's dead, and perhaps they could tell it was homicide based on something from the remains. So now they know they're looking at her now, not as someone who just walked away from her family, but as someone who was murdered. Who might be responsible? Oh, that boyfriend that we all suspected had something to do with it all along. That is him. And when they confronted him with, she's not just walked away, she's dead, he confessed. He legit confessed. And he's in jail. Yep. Now, in this case, this one's interesting. So this is one of those situations where there's DNA from a perpetrator at a crime scene, and they want to come up with what does this person look like. One really good thing about this is, say you've got, bless you, 30 people in your suspect pool, and your DNA phenotyping comes back and says this person has blue eyes. You can pretty much take everybody out that doesn't have blue eyes. It will help it helps detectives and investigators narrow that scope down. Like, it's definite, this person is definitely white. Okay, well, anybody that's not white, they're off the list. Now you've narrowed your list down remarkably. They were able to locate this dude. Now, this dude doesn't look like that picture, but that's because, bless you, when they create the phenotype, they have no idea how old the perpetrator is. They just know what their traits are. So they put everybody at roughly 30 years old. Everybody, because they don't know. They're dealing with an old person, a young person, they don't know. So they put everybody at roughly 30. Some of these you can go back and say, what did this guy, they will come out and show you a picture of that person at that same age after they've made the arrest. And they're usually freakishly accurate. They're really, really good. So one sec. So Parabon, since 2018, says they've solved more than 200 cases with the help of not only the phenotyping, but they also do forensic investigative genetic genealogy, which is the real cutting edge, the absolute cutting edge of right now of genetic forensic work. It is right there on the edge. So they don't want to get into a total number of cases because some are still active. They haven't made an arrest, although Parabon's done with their part of the job. They've delivered it. And next year, we're doing that lab that's gonna be a multi-week lab where you're gonna do the forensic investigative genetic genealogy, which is a big deal. Did you have something, Lucas?
So if someone took my DNA, could they see how it looked at like age 50?
I mean, maybe, I don't know if anybody's even geared their systems to age 50, they gear it to age 30, because that just seems to be. I mean, yeah, you could do that now without it though. You could, they have technology that can take you and age you. They've been doing that for years, like with missing children. They age them to, this child went missing at 7. This is probably how she would look at 15, probably how she would look at 25. They can do that just by knowing how features age. So now there are even more labs doing this kind of work, not just Parabon. Now we've got DNA International, Bodhi Technology, Othram, and Center for Human Identification.
When did it become like fully established?
I think it was early 2000s. I'm not sure of the exact year they came in, but they started coming around early mid 2010s is when they started really getting active in the scene. So they actually, I think they did call Barabon in the Bear Brook case, but that was towards the end of it. Yep. So this is how they do it, which is really pretty fascinating.
I'm Helen McRae, I'm the director of bioinformatics at Parabon Nano.
Guys.
And we do DNA phenotyping, so predicting a person's appearance, ancestry, all that stuff just from a DNA sample. We start from what we call genotype to phenotype data. That just means a big database where on thousands of samples, we have both information about their phenotypes, like say eye color. We have, are their eyes blue, green, brown, and we also have genotyped SNP data. So we've got about 1 million SNPs on each person and their genotype, so that's their AA.
So real quick, a SNP is a single nucleotide polymorphism. It's just a particular piece of a gene they're using that's unique to that particular phenotype.
At each site. And what we want to know is which of those SNPs is significant for eye color. We basically look through at each individual SNP and score how well does this data associate with the phenotype data. If people who have an AA at this SNP always have blue eyes, that would be a really strong association. It's never that simple, but that's what we're looking for. But the thing is, it actually gets much more complicated than that. If you look through the literature in genomics, you find what they call missing heritability, which means there are these traits that we know are genetic. They pass through families, they're very clearly heritable. But when we look across the genome, we don't actually find all the information to explain all the variation in that phenotype. So that's missing heritability. We're only explaining some of the heritability. And one idea for what might be missing is actually interactions among SNPs. So you might have a set of 5 SNPs that each of them individually has no effect on phenotype. But if you look at all 5 of them together, there are certain combinations of genotypes that have a significant effect. But if you've got 1 million and you need to look at 5 of the combinations, that's way more than you can ever test. I mean, you'd be doing calculations till the end of the universe. We don't have time for that. So what we've done is we've developed software that's called an evolutionary algorithm or a genetic algorithm. And so what it does is it starts from a random population and we're still doing scoring. We're looking at this combination of SNPs and scoring how well does this predict that phenotype. If it scores well, it gets to survive and breed and mutate, and we get to look all around it at other possible combinations of SNPs. If it scores poorly, then it dies off. It doesn't get searched around. So in that way, we're trying to explore the important parts of the space while not wasting the rest of our lives looking at the entire space.
So I'm going to pause her there because I do have other things to show you, but this full video is in your resources, right? So
those SNPs that we found.
Okay. Yep. This just happened last week. Like actually, I think not even, I think it was 6 days ago. Peaches doe and baby doe. So have you heard of Lisk? Anybody know of Lisk? Gilgo Beach. Lisk is Long Island serial killer, Gilgo Beach. So interesting story. Like literally there was a call, she was an escort at a guy's house at a party. She got freaked out. We don't really know why she got freaked out. Maybe they gave her something. Maybe she took something. I don't know. She ran out of the house. She started banging on neighbors' doors. She disappeared into the wetlands in Long Island around where this house was. Okay, the family and her, she had a driver. Some of these high-end escorts will have drivers to take you to houses so that it's a safety thing. She banged on his windows and then ran off and left him in the car. Anyway, when the police went looking for her eventually, they started finding other bodies. They do not believe she had anything to do with the rest of the bodies. They just found those bodies because they were looking for her. So they have found multiple bodies all along a stretch of road in Long Island called Gilgo Beach. And they are predominantly all female sex workers that were lost. People just thought they were lost. They weren't lost. They were dumped there. This lady, they didn't have a name for her. They couldn't identify her. So for the longest time, they call her Peaches because she has this little tattoo right here that looks like a peach. So that's what investigators called her, was Peaches. Now, Peaches, several years later, they found a baby. They kept finding more remains over a long time. They found remains and they found this baby. They genetically linked the baby to Peaches. So they knew it was Peaches and Peaches' baby, but they didn't know who Peaches was until last week. And we're talking a long time, a very long time. So 14 years apart between the mother and daughter, that's when they were found. And they think they may be linked to the Lisk, the Long Island serial killer or Gilgo Beach serial killer, who they have arrested recently, like 2 years ago. She is 26 years old. She was a military veteran. They believed that she was working at a medical office at the time. And so it says that she was a single mother at the time of her death, living in Brooklyn Sunset Park, served honorably as a veteran between 1993 and 1995. They found her in 1997, and then her daughter was found 14 years later. So this fellow here, Rex Heuermann, has been arrested for this. How did they catch him? Partly good old gumshoe. They went back and looked through everything and found that one of the girls who had gone missing, her roommates had described the man who she left with and his truck. And just so happened he drove a very, very specific truck, Chevy Avalanche. That's a highly identifiable vehicle. So they looked at everybody who drove dark colored Chevy Avalanches. There were very few. And then they started looking at their driver's license pictures based on the description they'd gotten. And this guy is huge. He's like 6'5", 300 pounds or something. He's a massive, massive man. He was described as an ogre by her roommates. So, and accurately, when you look at him, yep, totally see it. So how did they nail him? There was DNA on the victims. Definitely some left behind. There were hairs in one of the belts used to wrap one of the bodies that actually ended up in the end going back to his wife and daughter. Real nice. Pizza. He was eating a pizza in a pizza box, tossed the box. Trash is acceptable. They took it, swabbed it. That's your guy. That's how they got him. It was less than 2 years ago, and he's still not gone to trial because they keep adding bodies by linking them to him over time. Yeah. I don't have a picture of him. I don't want to glorify him, but you can look him up. He's a creepy looking dude. Rex Heuermann with 2 N's. Yep. I think they have charged him with 4 or 5, and there are 5 or 6 more that are likely his. Yeah, she got him. Ogre? Ogre. Yep. So what about the 4th Amendment? There he is. Yep.
Really good.
So what about the 4th Amendment? Your 4th Amendment is your what? Your right to privacy. Thank you. So say I have an account on Ancestry. I do, actually. Can law enforcement just go in and pull out my code?
They have.
They can't without warrants, and that's really hard to get. But here's how they do it. So private companies, they have to have a court order. That's really difficult. And it's hard to get enough. If you have enough evidence for a judge to give you a warrant to access someone's private genetic information, you probably have enough evidence to arrest the person to begin with. So they don't use those. They use open source DNA databases. And these are things like familytree.com or GEDmatch. These are open source. So the when you put your DNA into them, you take your file from Ancestry or 23andMe, and you take the file data and you transport it to GEDmatch, and you click up into law enforcement. And now your DNA is able to be used along the little chain of trying to determine who is who based on, it's not because they're looking for you, they might be looking for a second cousin twice removed from you, but they can use the percentage of DNA shared to build a tree to find the person. So the first big is called FIGG, Forensic Investigative Genetic Genealogy. Our victim in next year's FIGG case, his name is Hope Figg, spelled the same way. So the first big one, the first big publicized one was Joseph James DeAngelo, AKA East Area Rapist, Original Night Stalker, Golden State Killer. This guy went around California and terrorized people. You know who he was? Yeah.
So I know a lot.
Oh, this guy terrorized people. So this is a more recent one. It's this one, aside from the James DeAngelo, Joseph James DeAngelo, is this a new soon to be adjudicated, yep, that's him, soon to be adjudicated case of Brian Kohberger. So this is the Idaho massacre, the 4 college students who were in the house. It was 2 years ago, November, living in a house. It was a party house. And in the middle of the night, someone came in and literally slaughtered 4 of the people in the house.
Wow.
And it's very freaky because there's no, and here's the thing, he had no connection to any of them, and he was getting his PhD in criminal justice and stuff at the time, so he thought he knew what he was doing. Yeah, right. So how did they catch him? Well, they figured out who he was using tech. And then they nailed him, and he's dead to rights. He hasn't been to court yet, but he's dead to rights. And I'll tell you why he's dead to rights. So, meaning they're going to find him guilty. He's got no decent defense he can put forth. And here's why. So he went into the house in the middle of the night. He murdered 4 people. One of the roommates actually woke up, opened her bedroom door, and in terror stared as he walked past. Closed her door and went back in her room for hours before she came out and checked on. It was really weird. It's like she froze. She had like a psychological response where she straight froze. So anyway, they start looking who's been by here. Okay, you can't get away with any crime in a car these days because there are cameras everywhere. There are flock cameras like those ones at the entrances and exits of my neighborhood. If you start looking when you're driving down roads, any roads, public roads, you will see those flock cameras everywhere. They are tagging your license plate, and it's for the purpose of seeing who's where, when, okay? So all of those, so they have, and then they have cameras on businesses, yep. So cameras on businesses caught a white Hyundai Elantra going a certain direction and then coming back. And this was within the time frame they knew they were working on. The roommate gave them a rough time frame, plus, freakishly enough, the next door neighbor's Ring doorbell camera caught screams on the audio at a certain time, so they knew their time frame. So they saw the Elantra go by, they saw the Elantra come back, they're like, right in the right time frame. They start looking, eventually they find who the Elantra belongs to. So they think it may be him. They start looking into him, they check his phone. Guess what? Do you suppose he took his phone to the murder scene with him?
Almost.
No! He left it at home and turned it off during the window in which he committed the crime. That's not suspicious, right? If you look at someone's history of phone use and you see that they never turned it off, but they turned it off during this hour and a half,
Why wouldn't you just leave it on?
Smart person not only leaves it on, but has somebody else play with it or move it around. Nope, nope. So he thought he was outsmarting people. So then they found one piece of evidence at the crime scene, one piece of physical evidence they could use. Knife sheath found under one of the girls' bodies, and on the snap for the sheath was some touch DNA, skin cells from like closing the snap. So they went and hid outside his family's house and pulled he came out with like, he was sorting trash with rubber gloves and like carrying his, that's not suspicious either. Who does that? Pulled the trash. I think it was a water bottle in the trash. They tested not his DNA, his father's DNA. So that's investigative genetic genealogy. They can tell it's a son. So he's been arrested. He's going to go bye-bye, I'm pretty sure. So video surveillance of his Hyundai Elantra, cell phone geolocation. They also found he went by the house, I believe, 12 times over a 3-month period before he committed the crime. So he scoped it all on this phone. And then touch DNA on the knife sheath. And so here the fig was really easy. They only had to go one step. That's a father-son relationship. Easy peasy. Now, his lawyer tried the whole Fourth Amendment thing, but we've already hashed this out. This is known. We've already hashed this out. Trash, nope. Trash is permissible because it's abandoned, so it's fine, and you don't require a warrant for that, and you can even use more distant relationships. Your big names in FIG, your big names, your people on the cutting edge, your well-known people are Dr. Barbara Ray Venter and CeCe Moore. These are your big names. I think Venter works for Othram and CeCe Moore works for Parabon, or the other way around. If you're interested in learning more about Rex Heuermann's dirty deeds, there's this. And I warn you, it is not for the faint of heart. This is not somebody who just took people and then just killed them. He did a lot before he killed them. And it is not good. It is really gross. This is your item. And interestingly, every single person that when they went missing, like all the bodies they found, they're like, oh, this one went missing this week of this year of this month, you know, whatever that we know when she disappeared. Those were all times when his wife and children were out of town. Every time. So I'll come back to that if you want to see it, but here are your topics for your Unit 9 tests. And if you want, I'll go back. And if you want to hang for a few minutes, you can watch the Joseph James DeAngelo catch, and otherwise you're free to go. Yeah.
Grazie.
Yep. Yes.
For the final, is it gonna be cumulative?
Yes, it's the whole year. Do your review. Be really good about your review and you'll get everything on it. Yeah. We're back to political ads again. Oh, come on. Oh, no, don't do that. Do it. Yeah, you can put these on the counter if you want. That'll make it easier for everybody to get to.
American Family Insurance. Because for a limited time, buy one, take one is here, starting at just $14.99. So hurry on and enjoy your favorite creamy fettuccine Alfredo or our Prestige chicken.
For decades, California residents lived in fear of a notorious criminal lurking in the shadows and hiding on the sites of these selected criminals. Committing is free for kind of crime.
Yes, we'll be there.
Many cases. It wouldn't be until years later, with the advancement of technology, that Joseph James DeAngelo would finally be identified and linked to his crimes. In 2018, an ex-police officer who came from a military family was just an elderly neighbor to some. But little did anyone know that they were living in the presence of a monster with a grim past. One riddled with heinous and awful crimes.
Yeah, he's not
Born in New York, but moved to California as a teen, the root of his evil brewed somewhere along the line. As a kid, DeAngelo spent some of his time in Germany, where his father was stationed. While there, he witnessed a brutal crime against his younger sister, who was sexually assaulted by two soldiers. Whether the incident had any influence on the crimes he later commit is uncertain. But what is certain is that he too became a perpetrator of horrific acts. Between 1974 and 1979, while working as a cop, DeAngelo committed many crimes that he was never suspected of. A string of burglaries between 1974 and 1975, in which the suspect rummaged through women's underwear and stole small items, eventually led to that same person breaking into the home of Professor Klaus Snelling, where the perpetrator attempted to kidnap his teenage daughter. Snelling was shot and killed while confronting the offender, who would turn out to be DeAngelo, though police obviously did not know at the time. Even with DeAngelo still in the force, his extracurricular started attracting attention. By 1976, his crimes were being attributed to the East Area rapist, who was terrorizing Sacramento suburbs. In 1979, DeAngelo was fired from the police department for allegedly stealing a hammer and a can of dog repellent.
Yes.
After he had been fired, the police chief reported that his then 4-year-old daughter had seen a man's face outside of her window one night. Within the same year, DeAngelo was found guilty of shoplifting and was fined $100 and sentenced to probation. He sued the police force over his termination, which was later dropped. And as part of the legal proceedings, he had to do a mandatory therapy session in which he admitted to going to the chief's house with a gun with the intent to kill him. His then girlfriend, Bonnie Caldwell, who D'Angelo met at Sierra College and eventually became engaged to, has said that she became disturbed by some of his behaviors, such as illegal hunting and kicking the dog to death after it followed him while on a motorcycle ride. Caldwell may have ended her relationship with D'Angelo, but he wasn't exactly done with her. After the breakup, and with a gun in hand, he showed up at her parents' home where she was living, demanding that she go with him to Reno to get married. Caldwell told this to her father, who advised her to lock herself in the bathroom while he handled the situation. Eventually, her father got D'Angelo to leave. It would later be reported by many of his victims that he had broken into tears while attacking them, saying, "I hate you, Bobby." During D'Angelo's spree, there was a time where the activities of the East Area Rapist had seemingly diminished, but that's only because D'Angelo was on the move. However, a spree began emerging in Southern California that was attributed to the original Night Stalker, who had similar patterns in his attacks to the East Area Rapist. Initially, D'Angelo seemed to target women who were home alone or along with their children in suburban areas. Then he moved on to couples. He typically tied them in a knot, placing dishes on his back, cautioning him that if he hears the dishes, they'd both be killed. He would then sexually assault the woman in the house. Repeating this method of attack 21 times, he continued his reign of terror. Eventually, some of his victims fought back, some even escaped. After losing control of the horrendous scenes he was creating, D'Angelo then began to murder his victims. At the time, DNA technology was not available, so detectives used what they had, pieced together the patterns of the attacks. This led officials to piece together that the East Area Rapist might also be the original Night Stalker, who was murdering people in Southern California. Detective Larry Crompton of the Contra Costa County Sheriff's Department stated,
"He would not leave fingerprints, so we could not prove, other than his MO, that he was the same person."
Protect your family's dreams under our roof. American Family Insurance.
No prints and little known about his appearance, apart from minor details. No suspects were identified. It wouldn't be until many years later that the case would finally begin to crack as technological advances in DNA testing emerged. In mid-1990s, cold case investigator Detective Paul Holtz was working as a forensic scientist when he came across the case files and became obsessed with the case. By the early 2000s, DNA tests were available to confirm that the East Area Rapist and the original Night Stalker were the same person, but to no avail identifying a suspect. The late true crime writer Michelle McNamara dubbed him with the name the Golden State Killer in early 2013. She claimed that prior to giving him this name, the man lacked any sort of catchy name, despite having many associated with him, including the East Area Rapist or the original Night Stalker, which would shorten to ear/arms or ear arms. Even as of 2016, a $50,000 reward for any information that could lead to the arrest and conviction of the Golden State Killer was announced by the FBI. Detective Paul Holtz said of his interest in the Golden State Killer in particular,
"I was struck by the lengths this predator would go to instill fear in his victims. It was psychological terror."
Also saying that,
"For 44 years, law enforcement has been trying to solve this case. No other case has had more resources poured into it in the history of California. I was just stunned."
After speaking with a colleague about a 2002 case that had been solved using DNA in a genealogy website to identify a kidnapping victim, Detective Paul Holtz was inspired to look into this genetic method as a way of tracing the Golden State Killer. Over the years, many of the DNA samples from the case have been depleted, but a pathologist was able to duplicate an evidence kit that had been recovered from the case in 1980. Holtz obtained the sample he needed, and then found a website called GED Match, which is primarily used by people searching for possible distant family connections using their genetic information by accessing the website's database of nearly 1 million genetic profiles. Once the genetic material had been entered, the ensuing genealogy search was massive in scope. The initial DNA work was able to identify a few distant relatives, but not D'Angelo himself. The team then had 10 to 20 distant relatives of the killer, all roughly about third cousins. A team of 5 investigators built out intricate family trees, mostly on weekends and whenever they could steal a moment, because this investigation wasn't meant to be their full-time focus. The team spent months poring over census records, newspaper obituaries, grave site locators, and police and commercial databases. They first had to work backwards from every tree, linking all of the cousins to a common ancestor they shared with the killer, ultimately pinpointing D'Angelo's great-great-great-grandparents from the early 1800s. During this time, they built out about 25 family trees, with thousands of relatives from the 1800s to the present day. For scale, to locate the common ancestor, they had built out D'Angelo's family tree out to about 1,000 family members alone. They tried to locate men who would fall within the age range that could match the killer, who had any connections to Sacramento or other crime locations throughout California. Ultimately, they were able to narrow it down to 2 suspects. One of them was D'Angelo. Holmes was able to eliminate the other suspect through another DNA test from one of his relatives, leaving only D'Angelo. At the time, D'Angelo was 72 years old and retired, living in Citrus Heights, a suburb of Sacramento.
Right, cosmetics just came out with new shades of my favorite product, and I am obsessed. This is the Cloud Cosmetics. Brilliant. You don't even have to be a makeup artist.
Let's take about 30 seconds, and I instantly blow away.
I am not a morning person. I just make you what I know.
Uh-huh.
And they
Bowles learned that D'Angelo was a cop that had left the
have
force
like
in
their
his case.
heads or certain body parts, or like, they were all stuffed in burlap sacks and then thrown into what's essentially the marsh.
That's
that
what he
you
did.
can't get into.
Yeah, he talks a lot of.
Really
Yeah,
dense.
cadaver dogs were used in this case. Yeah.
So it took.
Investigators were able to match D'Angelo's DNA from the killers and taking samples from trash.
Some suburbs, the house he grew up in, but he turned out he was like a hoarder when I got his house.
Moreover, the success of finding the Golden Killer using genealogy and genetic technology has helped Spillman further advance in the justice.
Better next time.
One of the victim's brothers was able to successfully lobby California's government to expand the DNA collection.
Scope this area earlier, change the tires on the car and stuff like that, like he was real, and he's an architect.
Pled guilty to 13 counts of first-degree
Really
murder in special circumstances, such as murder committed during burglaries and sexual assaults, as well as 13 counts of kidnapping. According to Sacramento County Deputy District Attorney Amy Holliday, they came to an agreement, a plea deal, that if D'Angelo agreed to plead guilty on all charges, he would avoid the death penalty, though he would still serve a lifetime sentence without parole. Also by this plea deal, during his trial, prosecutors from the counties he had committed his crimes were able to lay out the nature of his crimes for the court in gruesome detail. A plea bargain also ensured the case didn't linger in court for years, and perhaps was a way for victims in this case to see justice promptly inflicted. In addition, D'Angelo had also been accused of committing over 50 rapes between 1975 and 1986. However, he could not be charged for them because of California's statute of limitations. When asked how he pleaded, he said guilty, and then when asked to acknowledge the sexual assaults and other crimes, he said, because of advances in DNA technology, and with the help of online genealogical catalogs, the infamous and monstrous James the Killer will finally be behind bars.
No, no, that will be Sink or Swim, I think.
Lawrence Lynn.
Yeah.
Last time I got some.
Yep. I'll get a phone from time to time. I think I was a very, very, very bad man.
Is he still in prison right now?
I think he's dead.
No.
He's
When
old.
was this?
Recently, within the last 10 years.
Oh, yeah.
I think he's dead.
Did the police kill him?
Did what?
Did the police kill him?
No, he went to court and everything, but he was very old when he got there. Alexa, is Joseph James D'Angelo dead?
From myheritage.com. Joseph passed away on month day 2022 at age 73.
Bye. Yeah, see you next week. Okay, excellent, thank you. See you later.